You would think that the person that should be giving the lecture on urothelial signaling has just stepped off the podium. But I'm not going to repeat anything that Laurie said. What I want to do, this is a workshop, so I want to throw out some very simple ideas and very simple things that I think that everybody who thinks about the bladder and thinks about the urothelium really should take account of, because much of it has been forgotten and much of it is actually leading people down the wrong path. So forgive me for being excessively simple about this, but we'll see where it takes us in the end. I think that should work. Help. That's my declaration. What we're interested in this series of lectures and part of the theme that we're running through the ICS this year is sensation and pain. But that's a very complicated system. And some years ago, Carl Eric and I came up with a simplification where we were thinking about the different systems in the lower urinary tract that might contribute to afferent flow, that might be involved in all of these systems. And we came up with the idea that these are the different systems that probably exist. There are C fibers, there are stretch receptors, and there's the role of the urothelium that Laurie has been the queen and proposed. And there are other systems that I've tried to convince people that do things as motor sensory system working through the ganglion. But today, in this 10, 15 minutes, I want to concentrate on some of my views of the urothelium. Laurie, you recognize this? This is what everybody recognizes. And it has, as Laurie has exquisitely shown, all of the elements that everybody thinks of whenever they consider the urothelium. The urothelium is a sensor. It creates lots of signals. Signals then go down. They activate afferent nerve fibers. And we have a very beautiful sensory system. But it's more complicated than that, even more than Laurie has suggested. I want to do that, show you these now. In my talk, I want to just focus on three particular aspects. One, I want to look at the specific innervation of the urothelium. I want to then consider complexities in the heterogeneity of it, not just in the complexity of it within cells, but complexities and heterogeneity within the different layers and within different regions. And I want to just throw something in at the end that says that we don't know everything yet, that there are other things yet to discover about the way the urothelium actually works and might contribute to sensation. Let me start with the innervation of the urothelium. This is a paper by Giorgio Gobella, published in 1998. And it's a piece of work that many people have forgotten. What he did, he was one of the first people to show the distribution of CGRP fibers in the wall of the rat bladder, and subsequently in other species as well. But this is the rat, and I'm going to concentrate on the rat. What Giorgio showed with CGRP antibody was that in the bladder, this is the urothelium as far as I can see it, excuse me. This is the urothelium, and you can see different areas of the urothelium. This is in the bladder base, and quite clearly in the bladder base, there is an enormous amount of immunoreactivity to CGRP fibers, putative afferents from the bladder in that region. However, in the lateral wall of the rat, guinea pig, and all of these other species, there are, in fact, no CGRP or any afferent nerve fibers whatsoever. If that is, and this is another section that actually shows going directly from the bladder neck up to the lateral wall here, and you can see some fibers, and then the absolute loss of suburothelial afferent nerve fibers. There are, of course, and I'll point out here, now CGRP fibers in the muscle, which are probably the stretch receptor ones that we were talking about. But this point is absolutely crucial. If there are no afferent fibers, then how does a urothelium in that region of the lateral wall contribute to sensation? What Laurie has probably been focusing on is the bladder base, where clearly there are afferent nerve fibers. But if you're going to take preparations of the bladder wall in the lateral wall or the dome area, and then do experiments that you're hypothesizing that the epithelial things you're going to do are linked to sensation, you've still got to answer the question, where are the nerves that these are going to talk to? And I think that that's a really fundamental hole that many people have forgotten. Now, let me leave that one there. This is some of my slides that just basically show this. We're concentrating on the lateral wall. Here is some of my immunohistochemistry showing we can see nerves around blood vessels, but there are no nerves. However, there are nerves in the muscle. The CGRP fibers are there, indicating that the lateral wall certainly does have some sensory function to play. So in this cartoon, I've shown the urothelium, the lamina propria, where there are no nerves, and here are the muscle nerves with the CGRP and the Vacht, which is the motor fiber. So this is a piece of microanatomy that we cannot deny, and we must take into account of what we do when we're doing experiments. If you go down to the bladder neck, the situation changes dramatically. Here you have an enormous innervation, which is fine. That's where all the signaling that Laurie might be talking about. But then there is another complication. It is not just simply a single type of afferent fiber. This is a section stained for CGRP and Vacht, vesicular acetylcholine transporter. And what it reveals is that there are multiple types of nerve fiber lying immediately below the urothelium. Now, are these motor? Are they sensory? We don't know. But they have all the characteristics of being sensory. But this then suggests that there are two types of sensory nerve fiber. If that's true, are they sensing different things? What are they doing? For the neurophysiologists amongst us, when you're recording from an afferent nerve fiber, which one are you recording from? Because that then means you've got to understand exactly, because they may be doing different things. They're in such close proximity, it also leads to the possibility of crosstalk between nerve fibers in the periphery, leading to all sorts of other integrations that might be going on. And in the pain field, this is called aphaptic transmission, whenever you get demyelination. So this is actually a well-known phenomenon of the possibility of crosstalk between nerve fibers in this region. So that's the bladder neck, and I'll point out that the majority of the fibers run parallel and are on the surface of the urothelium. However, if you go down to the distal urethra, you find that the distribution and pattern of the afferent nerve fibers is completely different. 20 to 30% of the nerve fibers now penetrate into the urothelium, and some of them, if you can see this revolving cartoon, I don't know if you can see it with the lights on. We can turn the light down to get some, that one, please. You can actually see that the fibers run through the epithelium and then float along the surface of the luminal aspect of the urothelium. You can just see the fibers. It's probably dark enough now to see the fibers running. This is the lumen here. This is the urothelium. This is the lamina propria. Here are the afferent nerve, the fibers, and here are some Vacht fibers penetrating, and here are some CGRP fibers penetrating. So the innervation has different aspects and different regions in association with the urothelium, suggesting possibly that they have different functions. And this is just a numerical summary of exactly that. Distribution of CGRP and Vacht, lateral wall, there's none. It increases as we go down the urethra, principally in the proximal urethra. This is a quantification of the number of fibers that penetrate into the urothelium, showing that there is something very interesting going on and different in the distal urethra as opposed to the proximal urethra and in the lateral wall. So this simple microanatomy must affect the way we think about sensation and outflow from the bladder. Now, that's one point. The next point is that we have a urothelium in the lateral wall. Well, what does it do? In the lateral wall, the urothelium, as we know, is a complex, multi-layered structure. And in the lateral wall, we have a basal layer, an intermediate layer, and in this particular stain, there's an umbrella cell layer up there that is just staining very faintly. This is a stain for nitric oxide COX and the nuclear stain DAPI. What this illustrates is that the different layers express different signals, and therefore these layers must have different functions. That must be it's a conclusion. However, in the proximal urethra, this complex signaling doesn't actually appear to take place in the rat or the guinea pig that I've looked at. So we're looking at a system here that is different when it's possibly communicating to the nerves than it is here when we're considering the interplay of signals between each other. So again, this puts another complexity, a heterogeneity in the type of cells in the epithelium within the same animal in different locations and regions. What does this region here of the urothelium do? Well, Laurie's shown quite nicely and said today that it releases signals. We know that. And the signals that are released can cross-talk with each other in a whole variety of ways. And here I've shown that ATP causes the release of prostaglandin, and here we've got acetylcholine also causes the release of prostaglandin. So there are complex interplays between signals in that region. But if there are no nerve fibers, what is this system doing? Well, it may be talking to the cells underneath, because the interstitial cells of the lamina propria are there also to receive signals. And one area that we've been interested in is their ability to signal and activate cytokine production. And this is leading us into a conclusion that this part of the bladder wall, if it's not doing an immediate sensory function, may be doing an immediate defense function. And what sort of evidence do we have for that? I'll come to that later. But what I also want to point out is something that I presented to the Society a little while ago, is that when you look very carefully for certain markers, they don't appear uniformly in the urothelium. They appear in localized areas that I've called hotspots. And these are small areas where you get specific expression. And in this particular example, it's cholinacetyltransferase. Here you see that the lamina propria is stained with vimentin. The urothelium goes all the way around the side, but it's only a couple of small patches that express cholinacetyltransferase. Now, many of the markers that we use to look at the urothelium actually also behave in this punctate way. And the example that I want to just rehearse a little bit more on is this molecular mechanism here, the protease activated receptor PAR1. And that's involved in sensing protease activity and then activating the cells. Activating the cells, let's leave it at that for the moment. And here you can see really quite nicely that there are cells that are expressing PAR1, the red, but clearly there are lots of areas of the urothelium where there is no PAR1. And it shows here in a very higher magnification, a bit fuzzy, that the PAR1 is expressed in the umbrella cells and nowhere else when it's expressed there. So the question is, what is this punctate element of the urothelium actually doing? Well, an indication to that may come from pathology models, and this is a cyclophosphamide prepared rat, and here is the control animal, and here is what you see typically in cyclophosphamide: damage to the urothelium and a huge expansion of the lamina propria. Under these conditions, the epithelial cells, now every single one of them expresses the PAR1. And we know that this is an important system with the expression because there is an antagonist, which you might hear about later, which actually stops the PAR1 system and stops it being overexpressed and stops it causing the damage as well. So we actually now can perhaps put a concept onto this as to what the lateral wall is doing. Maybe it's there to protect the bladder against something nasty coming to it. And what it does is it releases its signals to trigger all of the events that occur within the lamina propria to produce a protective situation. So this system is protective, but not in its immediate sense sensory. However, it does lead us to the possibility that this environment is now in close proximity to the muscle fibers, that if there is going to be sensation from a damaged area giving rise to pain, we must now bring the muscle into this. These CGRP fibers that are in the muscle are now in contact with some modified lamina propria. So maybe we should consider the concept of lamina propria as being the kind of sensor or trigger for sensation, not the urothelium in this particular location. The final thing I want to say in the last minutes that I've got left, if I can have the indulgence of the chairman, is I want to go back to the proximal urethra. In the proximal urethra, I've told you that there are different kinds of structure, Vacht fibers, and CGRP in green. What struck me was that when I looked at pictures like this, there is something incredibly odd. If you look at the high resolution picture down here of the red, which is a Vacht fiber, you can see that I can't see it there, but I hope you can see it. The Vacht fibers they have axons and they have varicosities. So what I did was simply to measure the diameter of the fibers and measure the diameter of the varicosities, and here you see the distribution. The diameter of the axon is just less than 0.5 micron, and this little bump here is due to the larger diameter varicosities. When you do the same thing for the CGRP fibers, you can see a CGRP fiber here with the axon and with a varicosity. When you measure the diameters, you get this distribution. The axons are, as you would expect, less than 0.5 micron. The vesicles, not the vesicles, the varicosities are about 1 micron. But there are structures in the lateral, in the proximal urethra that are very large indeed, with a median of about 3 microns. A structure that is CGRP positive with a diameter of 3 microns cannot be a nerve fiber. So what could they be? If you look very carefully at higher magnification, you find that these CGRP structures actually are nucleated, and they run along the base of the urothelium. If you measure the diameter of just cells that you can see the nucleus, you get the distribution that's at the far end of the CGRP situation. What I'd also like to point out to you is that these cells, which are big, which are obviously cells with a purpose, actually are intimately contacted by the Vacht fibers that seem to wrap around them. And if you play the video and if you don't get dizzy, you can actually see the Vacht fibers do actually wrap around these cells. So what are these cells? We don't know. They're not neurons because you don't expect to have nucleated cells underneath the epithelium in the proximal urethra. If you look at them in a little bit more detail, here we can see another one. You can see the nucleus of it. You can see this covering of CGRP dots and locations on the nuclear surface. But what this shows is that these cells also send processes into the urothelium. They are making contact. These contacts can be really quite complex, such that you've got many arborizations and small processes, thicker processes with other processes coming off. You can see one cell here with its nucleus and another cell here with its nucleus, giving the impression that this is a network of these cells, innervated in some way whatsoever. So, I've thrown a cat amongst the pigeons, I hope, by saying that we must now worry about where the innervation is in relation to the urothelium, because if there's no nerves, it can't be doing something sensory. The regions where we've got interesting things going on may have nothing to do with sensation, but maybe to do with repair or damage, which means it opens up all sorts of other interesting areas. And that's what we heard from Scott Huldren yesterday when we were looking at infection involved in these things. And there are still, I hope I've convinced you, interestingly new things still to be discovered that we have no idea what they do yet, but they're there and they need to be studied and understood in more detail. That's all I want to say. Thank you, okay?
Thank you. Thank you, Jim.